Okay, so we talk about these two important scientists. We talk about Robert Hooke. Now I'm going to move to Anton van Leeuwenhoek, who was the first person who saw the first living cells, and I'm going to talk a little bit about his life. So Anton van Leeuwenhoek is considered the father of the microscopy, and he's from Holland, and he made several discoveries at the microscopic level. And eventually, as all scientists, he took all his research and ended up publishing all his work. He sent a letter to the Royal Society, similar to what Robert Hooke did. He also published his work, and he had somebody that did all the drawings for him. Leeuwenhoek he started as an apprentice in a dry goods store where he was working with magnifying glasses, where to use to count the threads in cloth. And he was very interested in the topic, and he started working on getting better and better magnification and better resolution. And this very weird thing that you see here, this is one of the first microscopes that he created, and we still conserve some of these microscopes, and we can still use them just as a demonstration of what he was able to see using this very small, very primitive or early version of a microscope. He was the first person to describe bacteria, yeast, different type of algae, and he was the first one who described the diversity of a drop of water and the circulation of blood corpuscles in capillaries. And one important part of science is, as I said, is that the public knows all about what scientists know. So this is one of his quotes. Thought it my duty to put down my discovery on a paper, so all the ingenious people might be informed thereof. So again, to communicate to the public is very important. And what I'm going to try to show you what he was able to observe. As I said, Leeuwenhoek took samples of a pond water and viewed it through the microscope. The following slides, these are samples took from pond water, and I just wanted to show you what type of organisms what he was able to see. And some of these are algae, some of them are protozoa, but I assure you that if you look only in a drop of water, you might find a huge diversity of organisms. There is so much advances in microscopy. This is a competition companies that work in microscope do to try to show the diversity and the different movies that they can be done using microscopes. So just to remind you, Anton van Leeuwenhoek was able to publish his work, and in particular, we just watch all the diversity of organisms and small organisms in a pond of water. Why he got so interested in those topics? Because he was inspired by the work of Robert Hooke, in particular by the book of Micrographia. He made more than 500 microscopes, but only 10 of them survive to this day. And he tried to improve all these microscopes, and he was very skillful at grinding lenses. He was able to use this skill to magnify objects over 200 times. And similar to Robert Hooke, Leeuwenhoek hired an illustrator to create drawings of what he saw. Okay, so now we just talk about these two important scientists that were key for the invention of the microscope and the improvement of the microscope. Now I'm going to talk a bit about the cell theory and about microscopy. The work of Hooke and Leeuwenhoek was only the beginning of 2 centuries of observation and experimentation that allowed to establish the cell theory. So these two sciences were fundamental, but we need to wait almost 200 more of research and experimentation to establish the cell theory. And I'm just going to summarize a few of the important discoveries. In 1827, Robert Brown was able to describe a nucleus. He discovered a nucleus and he did a very careful description of that. Then in 1839, the second piece of of evidence that it was important to establish the cell theory is to these two German scientists, Jacob Sliden, a botanist, and Theodor Schramm, a zoologist. Independently, they found that both plants and animals, respectively, both have cells, and these two scientists are considered the co-founders of the cell theory. Now, the third component that is basic for the formation or for establishing the cell theory took place in 1855 when Rudolf Karl Virchow concluded that cells come from another cells, so that the cells don't spontaneously generate. They come from other cells that are dividing. And in 1855, Louis Pasteur found the evidence responsible for disapproving the doctrine of spontaneous generation. So from Leeuwenhoek to Pasteur and even to the present, there were two centers of discoveries and experimentation and observation that allow us to establish the cell theory. And the cell theory is a historic scientific theory that now is universally accepted, and it composed of three main principles. Here are the three principles if you want to take notes. The first principle is that all living organisms are made up of cells. The second one is that cells are the smallest units or the most basic building blocks of life. And finally, the third one, all cells come from pre-existing cells to the process of cell division. So we're going to talk a little bit more about each one of those ones. So as we said, the two German scientists, Sliden and Schramm, they found that both animals and plants have cells, but this is not the only example. Also bacteria, protozoa, unicellular organisms also have cells. Possibly the only exception is viruses, that they only have an envelope, a protein that surrounding them, and also on inside they have a nucleic acid. It could be DNA or it could be RNA, depending on the type of viruses. But still, we debate whether viruses can be considered living organisms or they are in between something that is alive or not. But in general, we can see that all living organisms are made up of cells. The second principle is that cells are the building blocks of life. As I said before, from bacteria to multicellular organisms, cells, they are fundamental for living organisms. In multicellular organisms, cells of one kind interact with other cells and form different type of tissues. So here you can have the adipose tissue, the nervous tissue, the bone tissue, the epithelial tissue, the connective tissue, and all of these tissues also interact with other type of tissues and form organs, and those at the same also can form system like the digestive system, and all the systems together where form a living organism. So from a simplest organism like a bacteria to a much more complex organism like all the different multicellular organisms, all of them are made up of building blocks that are cells. The third one, the last part of the cell theory, is that all cells come from our pre-existing cells to the process of cell division. The two German scientists, as I will show you in a following video, Schramm and Sliden, although they agree that both plant and animals are composed of cells, they have a dispute between this topic. For they were not sure. One of them was not sure that cells actually come from another cells. And at the end, as I said before, the two following scientists, Pasteur, showed that cells cannot spontaneously appear. They actually need mitosis, and a cell will come from another cell, and those cells will come from another cells, and those cells will produce more cells. Now I am going to show you a small video that summarizes the work of Robert Hooke, Anton van Leeuwenhoek, and a little bit of the cell theory.
Fast forward to the 1800s, where two German scientists discovered something that today we might find rather obvious, but helped tie together what we now know as the cell theory. The first scientist was Matthias Schleiden, a botanist who liked to study plants under a microscope. From his years of studying different plant species, it finally dawned on him that every single plant he had looked at were all made of cells. At the same time, on the other end of Germany was Theodor Schwann, a scientist who not only studied slides of animal cells under the microscope and got a special type of nerve cell named after him, but also invented rebreathers for firefighters and had a kickin' pair of sideburns. After studying animal cells for a while, he too came to the conclusion that all animals were made of cells. Immediately, he reached out via snail mail, as Twitter had yet to be invented, to other scientists working in the same field, and it was Schleiden who got back to him, and the two started working on the beginnings of the cell theory. A bone of contention arose between them, as for the last part of the cell theory, that cells come from pre-existing cells, Schleiden didn't exactly subscribe to that thought, as he swore cells came from free cell formation, where they just kind of spontaneously crystallized into existence. That's when another scientist, named Rudolf Virchow, stepped in with research showing that cells did come from other cells. Research that was actually, how to put it, borrowed without permission from a Jewish scientist by the name of Robert Remak, which led to two more feuding scientists. Thus, from teeth gunk to torquing off Newton, crystallization to Schwann cells, the cell theory came to be an important part of biology today. Some things we know about science today may seem boring.
So now we finished talking about these two important scientists. We talk about cell theory, and I'm going to tell you a little bit more about microscopy. Now we're going to talk about microscopy and different type of microscope that we currently use. Microscopy is defined as the field of using microscopes to view objects and areas of objects that cannot be seen with the naked eye. And they are fundamental to see different type of cells, different type of tissues, and we can now see even details in the cells, proteins, how the cells divide. We can see a lot of things. Now I'm going to talk about only 4 type of microscopes that are widely used, but there are so many more microscopes, and there is a combination and different styles of microscopes. For example, we're going to start with the light microscope, and this is a microscope that we have here, the light microscope. And this is the type of images that you get with a light microscope. And the key here of a light microscope is that the light come from here. Yeah, you have a visible light coming from there, and you can see or through the eyepieces in this area, or you can have a camera here that can help you to see that. And here you have a salmonella bacteria, and this is the type of images that you can get with a light microscope that again is using only visible light to see the sample. And here you are staining, this sample is stained. Here this sample is stained. Here this sample is stained with different chemicals that allow us to see the cells. The second microscope that I'm going to talk today is the electron microscope. An electron microscope gives you a much better resolution than a light microscope, and even that many other microscopes. Here you can see the same salmonella bacteria. Well, here you see the salmonella bacteria as all these dots. Here you can see it in much more details. The image is not colorful. We just with a software we gave the colors of this image, but generally the images are black and white. And here, of course, here you will have the eyepieces. You can see through here. These are all the controls. They look, as I said before, maybe old-fashioned, but these microscopes are still very useful. And the key here is that, well, the light microscope use visible light. Electron microscopes use electrons to create an image of the target. and in that way they can have higher magnification or resolving power than a normal light microscope. So visible light, here they use electrons to create the image. Now I'm going to talk about two additional type of microscopes, the fluorescent microscope and the confocal microscope. In the confocal microscope, this machine can be almost more than $300,000 or $400,000. So they don't look very expensive, but they can be very expensive. And why these two type of microscopes are so different from the previous, from the light and from the electron microscope? Because they what they are doing is to irradiate the specimen using a different type of light, and this type of light will emit a fluorescence. So I will show you a few pictures in a second, but the idea is that, well, the visible microscope, you will have visible light. In the electron microscope, you create the image using electrons. Here you need to irradiate the specimen with a specific wave length, and this will allow to produce a different fluorescence. And here you can have the UV light that it will irradiate the sample, and it will cause the excitement of the light. And here in the confocal microscope, you have a very similar story, but this one will allow you to make 3D pictures, to have a 3D resolution, because it allows you to remove light out of focus. And maybe it's much clearer if I show you like this. This is the fluorescent microscope, and this is the confocal microscope. If you see that these ones are the same pictures, well, these two are the same picture, and these two are the same picture, and A and D are the same picture, B and C and F are the same picture. The only difference is that the confocal microscope removed the light out of focus using a pinhole, and this will allow to make the image much sharper. And to finish, I just want to tell you, just to remind you, we talk about four type of microscopes: the light microscope that use visible light, the electron microscope that use electrons to target the image, and finally these two microscopes that use fluorescence by irradiating the sample with a specific wavelength, and as a result of that it produces this type of fluorescence as you can see here. And the only difference is that the confocal microscope will make the image much sharper. You will take much longer to take a picture in a confocal microscope because it's removing the background, all the light out of focus, but both are very useful. To finish, I want to show you some pictures, comparing some light microscopy pictures that we have in the past, or drawing that we have in the past, with pictures that we can get with this type of confocal microscopes. And even I will show you some type of experiments that we can do now, not only using focal microscope, but they can use also lasers to do experiments that it can help us to understand much better the mechanisms of development of different organisms. So, well, in the past we were only using the light microscope, and we only have drawings in the past. Now we have different stainings that allow us to see different tissues and different type of organelles. This is, for example, single cell protozoa, and now we can see in much more detail. This is another example. Maybe you have seen this in a light microscope. This is a tardigrade. Here again we can have different stainings to see different structures that can help us to differentiate much better the different structures of very small organisms. Finally, this is a drawing of an embryo of a turtle. Again, now with different type of staining and different type of microscopy, we have not even improved the resolution, but also we can label different structure. This is an embryo of a turtle. I'm not going to get into a lot of detail what the red and the purple means. That is expression of different genes, but just for this purpose, I just wanted to show you that. Finally, I just wanted to show you, to conclude, I always love to show you movies. This is a movie of a fruit fly during embryogenesis. Here you find one tissue, and here you find another tissue. And if let me play it again, and then they are closing and they are attaching together. And this process is called dorsal closure. You can see tiny arms, tiny fingers attaching to each other. And this process is very important to study how wound repaired, how injuries are repaired in all the tissues. And but we are not only interested in trying to understand microscope now. They not only allow us to study how tissues develop, but also to go from a transition from observation to experimentation. So, for example, here you can use a laser to cut the tissue and to see how it heals. Here in this tissue you only have water, while in the one in the right you have a mutation, and you can see that the tissue is not healing very well in the right, while in the left was healing much faster. So again, right now microscope not only allow us to make observations, but also allow us to make a microscope, and these are just a few examples. And there are many more microscopes, and there are many that allow us to make much more experiments. But I'm not going to get into much more details in this class.